very much, Elena. Thank you, and it's been an honor and a pleasure also to have you here with us, and thank you also for your amazing presentation that for us is very important, also because the next, you can say, part of the conference will deal with the topics that also you have talked about. And thank you also for stressing on the concept of epigenomics. That is something that is a prominent and we can say innovative field of research that as One Health we want to encounter and promote. Thank you very much, Elena. So now I'll give the floor to the next session. That will be the first dimension that we identify as, we can say, a component of the One Health framework, that is the environmental and ecological dimension. Now we will give the space to the keynotes of the session and to the authors that have presented to the call for papers. So why the environmental dimension? I think the response could be trivial. Of course, the One Health stands properly to understand that the health of humans is also connected to the health of environment and ecosystems. So I kindly ask to the chairs of the session to join the floor. One of the chairs is already here, Professor Margherita Ferrante, and I'll kindly ask to join also Professor J. Alivieri. Okay. Yeah.
Okay, first of all, I want to ask to you if you are the question for the previous speaker. You have the question for Amina, or for Marcella Trombetta, for Elena Codina? No, all is very clear, and the talk are very, very interesting and very worrying for us that are shown us how much we are distant from, far from the result that we have to reach very soon. Okay, now we start with the next session. The section is dedicated to the topic unleashing microscale understandings of One Health. And we have three speakers, three very important speakers: Mohammed Banni from Hagen Institute of Biotechnology, Monastir, Professor Alberto Rizzotti from University of Genova, and Professor Giovanni Maga, Director of the Department of Biomedical Science, CNR Italy. And the three speakers can help us to understand these important topics for the knowing the understanding of One Health concept.
Welcome all you in Barcelona. I'm very happy to meet you, all you, in this convention. I have the honor and I'm very happy to present Mohammed Banni, a friend and a very valuable scientist, specialized in microplastics effects on environment and health, human health, and only is a very, very big scientist on the topic. Thank you for your presence here, and the floor is yours.
Thank you. Thank you very much, Jaya, for this very kind presentation. I think that you are overestimating my my scientific, let's say, value, but let's say thank you very much. So what I will present today is the microplastic from the environment to the human health. And it will be something that will, let's say, provide some key information about the how far microplastic could be dangerous for many aspects linked to the human health, but not only the human health, for the One Health in one concept. So, okay, the next slide. I will begin with this very common list of sustainable development goals that have been established and then maintained in all the aspects related to the One Health. Microplastic, I think, is everywhere here. So if we focus a little bit on the presence and the effect of microplastic. They are everywhere. Gender equality, for example, what will be the impact of microplastic on this? We can have some response. Also, the good health and well-being, quality education, because through education, we will probably mitigate this problem. So this is just to start this presentation. The next will be a very interesting paper that have been recently published on a top journal of environmental sciences. And in this paper, my group demonstrated the negative impact of microplastic on the reproduction of mussels, so link it to what we call the biodiversity decline. Unfortunately, these data are really very worrying about the possibility of extinction of species. I'm whitening my words when I say extinction, because through reproduction, you will limit the, from one side, the biodiversity, and then the representation of one species in one environment. Also, I will go with the evidence. And here we have also two papers that related the microplastic role in carrying harmful substance in the environment. And in this case, it is one hydrocarbon, and the second is pathogenic bacteria and virus. So microplastic is not only dangerous and harmful by itself, but through carrying a lot what we call the plastisphere. And the plastisphere will be also chemical and biological components. The last slide with this, let's say, publication also, and these are very interesting, performed with the Margherita and Jaya group, where we demonstrated that microplastic could reach human health through the web food. The first with the fruit and vegetables, something that was really unexpected, and the second through the edible fish in the Mediterranean. So that's it. I mean, this very small presentation was to show how microplastic can be present everywhere and can impact our daily life. So the idea through this experiment is to show that microplastic, not only microplastic, but antibiotics that are very present in chicken rare, and this could affect one key organism in the food web. And here we have provided some we will try to provide some information about what will happen in the liver of this animal once exposed to microplastic and an antibiotic, and in this case is oxytetracycline. So the data are performed on the liver of this animal, and what we can see here is that after 7 days of exposure, we have parameter related to the cell death in the liver of this animal. And the tunnel assay, a key parameter showing the cell death through apoptosis, clearly indicate here that you have microplastic contribution to the cell death, but microplastic with antibiotic would be something more important. Key parameters like cytochrome C, but also P53 and the BAX BCL2 are also were also investigated, indicating the same path and the same probably issue in the cell death of these cells belonging to this animal exposed to this kind of contaminants. So what will be the message here? The message here is that in a very conventional and practical issue, when we are dealing with chicken in farming system, microplastic would be a key element present in their waters and also in their food. And this will directly impact the health status of these animals. Unfortunately, we have also other data that I'm not presenting here related to the accumulation of microplastic in the edible part of the animal, I mean the flesh, and they are really very worrying. So the idea now is once we will consume these animals, because it is a common thing, what will happen to humans if we will maintain the same concentration that will be found in this animal. So we decided to work on what we call the stem cells, the human stem cells, that will differentiate further to provide our bones or the fat tissues. And these stem cells, we have decided to work on the bone marrow stem cells and the adipose tissue also as key, let's say, starting tissues. The idea is not to work on cancer cells, cell lines, because they are already, let's say, modified, and we risk to have some controversial data. But on stem cells, the issue is stronger, because we are dealing with also all kinds of human beings, from children to adults. So in one word, these cells would provide, through differentiations, osteocyte, chondrocyte, or adipocytes in a proportion that has been maintained, that is maintained. But the idea is, will this proportion of differentiation will be maintained under microplastic exposure or shifted toward a specific tissue? I mean, instead of giving bones, will it be possible to have more fat tissue than bones? And the response was really shocking. So the first data here with the bone marrow or the adipocytes will be under exposure of microplastic. The first test is the cycling cells. I mean, the cells undergoing cyclic divisions. In control, it is very normal, but under polyethylene terephthalate and polyethylene terephthalate of two sizes, 1 and 2.6 micrometers, we have a drastic decrease of the cell cycling. I mean, the cells are not undergoing normal division, and this is very worrying. The second is the same with the three, or the kind of cycle steps, sorry. The phase S, which is the most important in the synthesis of DNA, and here we can clearly show that under microplastic exposure, this is drastically changed and modified. Sorry, see. This is the senescence. I mean, if this size cells under microplastic going through death or through normal cycling life to provide this endpoint osteoblast or chondrocyte. Or so the problem is here that the majority of the cells under microplastic are going under senescence. I mean the death, and not probably provide correct cells. Yes, apoptosis is also investigated, and as you can see here, we have not only senescence that is worrying, but also apoptosis, and these cells are undergoing also a drastic apoptotic level respect to control. Okay, these are the same, but under protein, let's say, point of view, and we had investigated all the protein involved on all these pathways that I have just mentioned now. Okay, the effect of polyethylene terephthalate. Let me just remember that the polyethylene terephthalate is the main plastic polymer used for water bottles. So, and here we have a team that have worked drastically on this issue and demonstrated the presence of a huge number of particles in drinking water when you are drinking water into bottles, plastic bottles, let's say. So here the idea is to show the decrease of the number of colonies formed under microplastic exposure. Colony formed means also the expansion and the division of the cells. So we are really joining the same issue under the same conditions. And here it is a manifestation and the representation of the typology of the cell. I mean, after division, those who have escaped the senescence or the apoptosis, which will be their endpoint? They will be either osteoblastic or adipocytes. So a specific dyeing or specific dye will reveal the type of the cell. And here you have the red oil, which is very representative of the adipocytes. Alizarin also, and safranin is representing specific type of cells. So yes, the cell ability to differentiate into adipo, osteo, and chondrocytes, and here the most important and shocking one is this one. Cells are undergoing more pronounced differentiation into adipocyte. In one word, this is a first evidence that stem cells under microplastic, they are undergoing a shift to adipocyte rather to chondrocyte or osteoblasts. So this could explain in somehow the obesity in some case of children. But we need to go with further details with this subject. Okay, I will go, of course, the ROS, but I will try to go more rapidly with this second part. That will be the first attempt, really the first attempt to mitigate the microplastic, because mitigation strategy of microplastic are often, let's say, undertaken at the basal level. Let's reduce the plastic, let's reduce the plastic entry into the food web, etc., etc. But once inside the organism, what will be the solution? And the key word that we have identified through the microplastic effects and how microplastic could change the cell physiology is under the promotion of oxidative stress. This is one aspect, but also the promotion of DNA alteration. DNA alteration physically through the division of cells, because microplastic have this small size that could interfere with division. So how to mitigate? Our solution up now is melatonin. Melatonin, considered as, let's say, safe substances that we are producing ourselves in a certain path, controlling the night and the day issue for humans. Melatonin is responsible, is highly produced in human when we are sleeping. So it is controlling our, let's say, night and day life. But melatonin is also reported to mitigate a lot of contaminants such as cadmium and many other pollutants that interfere with cell biology. So the idea here is to put the same condition, but introducing melatonin either 6 hours before the exposure or during the exposure. So, and this is the very astonishing, let's say, response. In terms of apoptosis, we have this apoptosis and necrosis, but let's focus on necrosis. In control cells, there is no necrosis, quite no necrosis. With melatonin, of course not, but with PET1 and PET2.6, the necrosis of cells are really increasing drastically. Melatonin, for sure, do not decrease it PET-induced necrosis, but decrease it PET with size of 2.6, increasing necrosis. So this is the first evidence of the potential use of melatonin to mitigate the plastic effects. Also in terms of senescence and in terms of ROS production. So let's see here in terms of senescence, and you have the polyethylene terephthalate increased drastically, also the senescence, and the melatonin after 6 hours reduced somehow. I'm sorry not providing some statistical issue, because these data were collected only on Monday, yesterday, in 2 days. So I'm really very excited to present them, but without having any influence in terms of statistical analysis, but they look fine. So that's why I decided to present them. And the most important one is the ROS, reproduction of oxidative stress through melatonin reduction is really incredible for microplastic with that 1 micrometer. So the idea now, I know that these are very preliminary data, but I think that we are in presence of a potential use of a natural substance to mitigate the presence of microplastic and their effect on human cells. I think that we will not be able to reduce the ability of the microplastic to enter the food web because of what we know about their disponibility, availability, their presence, etc., etc. But at least for the next 20, 30, let's say 30 years, I'm not sure that with all the effort that we will perform, we will be able to reduce their presence in the environment, because we are now under the presence of microplastic that has been introduced probably 20 or 30 years ago. But if we start now, probably we will say that in next generation, microplastic will be reduced. The solution we are providing here is within the problem that will not solve at the let's say at the basal level, but at least we can mitigate it at cellular level. This is a first evidence, and I would really stress this fact. We have to deal with microplastic, not only try to mitigate them, because actually it is, let's say, a nonsense to mitigate what is really existing now. Probably what will be introduced in the future, I agree, but actually we are living under the pressure of what has been introduced into the environment probably 20 or 30 or 40 years ago. So thank you very much for your kind, I will speed up because, and I would like to thank all my team that is really working hard to provide this data, either in SUS or through my collaboration with the University of Catania, the University of Luigi Vanvitelli in Naples, and also we have a very interesting work that is now under publication on the effect of microplastic on bacterial population in gut. I mean, in living organisms such as seaworm and also earthworms, so the metagenomic approach in response to microplastic. And there, the idea that I have launched yesterday, probably in some situation we need also to take advantage of some problems. And one key point that we have identified with our colleague from the University Cattolica of Milan, Professor Edoardo Pugliesi, that under microplastic, seaworms and earthworms are producing antibiotics. And they are very small, let's say, peptides that can be used in health, in human health. So these are really innovation. Unfortunately, they are under stress of microplastic, but this is the response of living organisms. So thank you very much.
Thank you, Mohammed. Very interesting presentation on the problem of very great concern for the environment, for the human health, on the light of the studies that are present now in the global literature, but also on the light of your studies and our studies that are a very great pleasure to make with you. There are some questions for Mohammed. Okay.
Thank you.
Thank you, Mohammed. Question. Please.
A moment, a microphone. Yeah? Can you use my microphone?
Currently, with online shopping, after COVID increasing and people buying more plastics around the world, how do you feel that perhaps even scientists or even the community, what can we do to make better choices concerning this issue? Or how can we face this issue as scientists concerning the large amount of plastics that are going to be consumed in the next years?
Thank you very much for this very crucial question. I was asked a similar question about what will be the role of scientists in mitigating microplastic, not only in their lab, but also through the dissemination of information and the dissemination of information. And one of the questions was, wherever the lobbying is existing behind the plastic production would be stronger than what our effort as scientists will be. And I remember that Professor Galgani from France responded to this question saying that manufacturing plastic is providing money for those who are producing plastic. And this is their job, so they will not abdict. But if there is no need for plastic, these guys will not produce plastic. I mean, in one word, if people are continuing consuming water in a plastic bottle of water, it is stupid to think that who are producing plastic will stop producing plastic. Probably they will shift somehow, but it is our demand, our comfort as consumers who is pushing people to produce more and more plastic. It is our demand on masks during the COVID-19 who pushed people to produce because it is a market for them. So I think that microplastic, not microplastic, but plastic in general, could be considered as a common worrying issue for all human beings. And we should take one decision. Probably we will not adhere, all of us, to this decision to say, let's reduce the consumption, the consuming of plastic. Unfortunately, there is no conviction in all the countries around the Mediterranean, at least, to stop producing at least one step. Stop producing plastic, mono-use plastic, the plastic with one use. Why we are continuing producing this kind of plastic? I know that in Canada it is under law. You are forbidden to produce, to use, and to commercialize this kind of plastic. So probably it should be something that all of us should work behind it. And I see just one issue to succeed in this so hard, let's say, thing, through education. I believe that from children we can produce for the next year a better world. So we need to stress in our research programs, and it is one thing that I'm doing through my research programs, to put clearly that a good budget must go to the increasing awareness with children. And this will be something that will be successful, not now, but in the future. Sorry. I would not repeat it, but I did.
Thank you, Mohammed.
Thank you. Thank you for the very informative presentation. Maybe I didn't catch up all the details, but I just want to comment on the question about how to make it work. I don't know if you're aware, in March 2022, the UNEP, the UN Environment Program General Assembly have adopted a resolution to have a legally binding treaty on plastic pollution that is being now negotiated. So it was just to comment that there is something that will be legally binding, but my question to you, you were also talking about some countries that have taken different steps. Would you think that having something at global level that is it working?
Open
Oh,
the microphone.
On.
Is it working now?
Okay.
Oh, sorry. Should I repeat?
Okay. Okay.
No, please, okay. No, I was just mentioning that in March 2022, at the UN Environment Program General Assembly, there was a resolution that was adopted to have a legally binding instrument on plastic pollution. This treaty now, there is an inter-governing body that is being drafting this treaty. So the question I have is that, do you think that having a treaty that would be legally binding will convince, or I think if there are legal consequences to the consumption or to the use of plastic, my question to you, do you think it's going to help all the work you're doing? Thank you.
Thank you very much for your question. I think that all the initiative will be very welcome, because it is only through binding all the initiative that we could succeed to reach an end point. But let me just provide one personal, let's say, experience with this kind of initiatives. And I had a project, an NECBC MED project, dealing with microplastic in the Mediterranean with colleagues from Italy, from Lebanon, and also, yes, Lebanon and Italy, and Tunisia. And during this program, we tried to be more than scientists and to provide some issue dealing with policymakers and yes, policymakers. One of the suggestions that has been proposed at the end of this program is to impose a certain kind of tax on plastic that go to research on plastic. So, I mean, in Tunisia, at least, we have this very interesting law that on one bottle of plastic, there is a tax that will go to the collection of plastic after the use. And it is provided to the local communities to do this job. And it will be more efficient, according to me, to impose also one small quotation of tax to increase the awareness on the use of plastic. That will go through I don't know how exactly we could use this money, but probably to increase awareness in a target population like children in schools, like etc. My conviction is that because the idea to say to a person, stop smoking because smoking is leading to death. Nobody will, but if you will vehiculate this message to children, 10 years old children, it will be more impacting. So let's work in the next generations, because in my idea, actually, you can increase awareness and have a positive impact on the increasing of awareness through these low age levels that will be really effective in 10 years. So this is my idea. It is not the correct one, I agree, but.
Thank you.
Any other questions? Yes. Oh, yes.
Thank you. Outstanding presentation. Thank you very much. So I have just a curiosity. Did you check some effect coming from bioplastics until now? And the second one, I enjoyed very much to see all the data in short time, wonderful data. But in my opinion, can be good also to show some data about the repair of DNA. Did you check already?
The DNA repair?
Repair of DNA.
Yes.
You already did?
Yes.
Okay. So no collaboration for this step.
No,
I was asking to share some knowledge about. Okay.
Thank you very much, and thank you very much for your kind words. I will respond to the first and the second question. So the first question is the DNA repair, and
if you check already the effect coming.
no, the bioplastic. Yeah, the bioplastic. So I have a drastic position within bioplastic, because people think that using bioplastic will be a solution. It is not. It is not. Because at the end, bioplastic will be very attractive to pathogenic bacteria once, because bioplastic is based on sugar. And this will be a carbon source for the development of bacteria. And indeed, our results with the metagenomic analysis on bacterial colonization are really very worrying when you are talking about PLA. And the other issue with bioplastic, people think wrongly that bioplastic would be more degradable. It is not true, because through our analysis, if you will take PLA, and unfortunately the lobbying behind is pushing in the other side, PLA will be degraded in polylactic acid, and you will find in the medium a very high pH. So if you are thinking to produce compost, in this compost, nothing will go. So these are data that I have stolen to Eduardo Pugliese, but it is very nice to communicate with the other scientists. For the DNA repair, of course, yes. And the first paper that we have published was with my PhD student Elif on the ability of muscle hemolymph to repair DNA under microplastic exposure. And unfortunately, it is the apoptosis is the one that will win at the end, because repairing DNA under microplastic, the DNA will undergo macro alterations, not micro alteration. A DNA strand break, single strand break. You will have a very pronounced comet issue. You will have a very pronounced number of micronucleus, and this kind of alteration will not allow the DNA repair. At the contrary, if they will escape to the cell death, it will be promoting cancers in certain cases.
I was thinking about adiposylation enzyme to check.
Yes.
Yes.
I would maintain a certain kind of hope in the ability of the cell to go further.
That are clear already.
I would be in favor of a positive reparation ability, and the ability of the cell to cope with stress is not always successful. So I would, let's say, not rely on the ability of the cell to mitigate such problem. It would be in the case of chemicals, benzoypyrin, reparation is possible. But don't forget that humans are equipped with the genetic and epigenetic capabilities. But other organisms, if we are thinking about environment and about the what we call the biodiversity decline. For example, mussels, bivalves, are not equipped with DNA strategies to mitigate some contaminants like benzoypyrin. They are not able to make oxidation. So it may accumulate, affect, and you have this kind of long-term effects.
Thank you so much.
I will try to make it as quick as possible.
No.
Thank you so much for the presentation. I was just wondering about the last point that you raised about the synthesis of antibiotics by worms after the exposure of microplastic.
Yeah.
I can obviously see the potential, but I'm also considering the issues that we have with antibiotic resistance.
Yes.
I'm afraid that an increase of antibiotics in the environment and therefore the insert of antibiotics in the food chain might lead to further problems down the line.
I totally agree with you. It was just in the line of one comment that I did yesterday during the launch of the One Health conference. I mean, I just wanted to say that under pressure of microplastics, certain organisms, and mainly in their in combination with their associated bacteria, mainly in the gut for animals and for us in the intestine, would produce a certain kind of molecules to mitigate microplastic. But indeed, they are not mitigating microplastic. They are reducing bacterial activity. So that's why we consider them as antibiotics. But totally agree with you that the increase of the number of antibiotics in the environment with the issue of antibiotic resistance. And imagine, without introducing any new antibiotics, you will have antibiotics in the environment under microplastic. So this is a very big issue. And I wanted to stress it.
Yeah, thank you. There are some very cool studies about antibiotic resistance being caused by non-antibiotics.
Yeah.
So it's something that it's becoming more
and I'm
more
sure I was about to provide this to introduce some information in my lecture, but unfortunately these are really very, very innovative data, and I would have the consent of the colleagues with whom I worked to present them. So,
thank you so much.
Thank you.
Do we have any other questions from the medical perspective as well?
No.
Okay, thank you.
Thank you. Thank you very much.